So today we're going to talk about DNA and RNA. So this comes from your chapter 12, but it's my variation of chapter 12. So in the 20th century, it was very exciting because that's when scientists discovered that DNA, or deoxyribonucleic acid, was a thing that had basically all the information for living organisms. Of course, with that discovery, then there was like a race to figure out more about DNA. So what did it do? How do you get more of it? That kind of thing. Early on, all scientists knew was that DNA was able to store information pertaining to development. They knew that it could be replicated even, and that it could undergo rare changes called mutation. So there was still a whole lot to know about it. Back then, Frederick Griffith actually was investigating something different. He was looking at how dangerous this strep strain could be. But what he ended up accidentally finding out was that the dangerous component of this strep could be passed to a non-dangerous strep and made dangerous. So it actually had a transformation. And so then further research done by Avery, they figured out that DNA was the thing that transformed that strain from being non-harmful to harmful. So what Griffith did was basically inject a deadly strain of the strep into the mice. They died, okay? He injected a non-deadly, non-harmful strain into the mice, and they were okay. He then he killed the strep strain, and since the strep was heat killed, they lived. But when he injected the heat-killed strain into the mice along with a living strain, the living strain accidentally incorporated some of the deadly strain, and therefore the mice died. So what we know about DNA now is a whole lot more. We know that DNA is a nucleic acid, and we know RNA is a nucleic acid. So DNA is in the nucleus only, RNA is found in the nucleus and the cytoplasm. And building blocks of DNA are actually nucleotides, all joined together. So a nucleotide is made up of a phosphate group, a sugar, which is deoxyribose for DNA and ribose for RNA, and then a nitrogen base. So the sugar and phosphate are the same in every nucleotide of DNA. It's always the same sugar, same phosphate, okay? But there are different bases, and so the bases are the things that change. So there are 4 different bases for DNA. There's adenine and guanine, and there's cytosine and thymine. Adenine and guanine are the bigger bases, and so those are purines, and the smaller bases are the pyrimidines. So those nucleotide building blocks bond together to make long strands, and it looks like a whole lot like sugar, phosphate, sugar, phosphate, sugar, phosphate, with the bases like sticking out on the edges. So DNA is double-stranded, RNA is single-stranded, DNA has thymine, and RNA actually has a different nitrogenous base called uracil. So we say good old Watson and Crick because we know that we have seen the movie about Rosalind Franklin, and we know that Watson and Crick actually got their information about what DNA looks like based on Rosalind Franklin's work. But because of the model that was built, we know that that DNA does look like a double helix, and it appears to look like a twisted ladder. And so the sugar phosphate make up the sides, and then those hydrogen bonded bases make up the rungs. And because of their work, where they built the model, Watson and Crick actually won a Nobel Prize in 1962. So basically, the function of DNA is to store the genetic information for everything that makes us us. And it's made up of 2 strands that match up with each other, and that's called base pairing. And so there's adenine, guanine, and thymine and cytosine. So the adenine and guanine are the purines, The way I remember that is like chemistry, Ag is silver, and so I remember pure silver, which is kind of hokey, but it works. And then thymine and cytosine are the pyrimidines. And so there's complementary base pairing, and the rules don't change. So adenine always bonds with thymine, and guanine always bonds with cytosine when DNA bonds with DNA. So this is the component that makes up a nucleotide. So there's the phosphate, the sugar, and the nitrogen base. And so we know if we're going to make complementary base pairs with this other strand, we would know that it would look a whole lot like this. G to C, C to G, T to A, T to A to T, and C to G. And so if you lay DNA out flat, it really does look like a ladder. So RNA has a huge role in the job that DNA can do. So DNA doesn't leave the nucleus, so it has to have a messenger molecule that goes from the nucleus to the cytoplasm. And so basically, because we know back in the day when we were learning about cells, we remember that DNA is in the nucleus and it doesn't leave the nucleus, it's way too big. And we also remember that DNA isn't even the organelle that makes proteins anyway. And we know that anything that has to do with you is essentially a protein being expressed in a way. And so we remember that ribosomes are actually the things that make proteins. And so somehow DNA that has a recipe to make every kind of protein our body needs is stuck in the nucleus. And so we have to get the memo that's transcribed in the order that DNA's nucleotides are arranged in. We got to get that memo over to a ribosome. So RNA is basically the thing that does that. So it gets the information from DNA, takes that information over to a ribosome so that a ribosome can then make the correct protein. So RNA has a sugar, which is ribose instead of deoxyribose. It has a phosphate, and it has a nitrogen base. And RNA can make pairs with DNA or other RNA, which is really important with transcription and translation. And so when RNA pairs with RNA, cytosine pairs with guanine, adenine pairs with uracil, and pay attention, RNA has uracil instead of thymine. Now RNA can make pairs with DNA, and that's basically how RNA gets the memo, if you recall, to take that information from DNA. So basically, RNA gets the memo from DNA by making a complementary base pair with DNA and then takes that memo to a ribosome. So cytosine pairs with guanine and vice versa. The uracil of RNA pairs with the adenine of DNA, and the adenine of RNA pairs with the thymine of DNA. So if I would have a DNA strand and I was going to make a complementary base pair with an RNA strand, I would know that C goes with G, A goes with U, G goes with C, and T goes with A.